In this video, we're going to consider the ways in which molecules get into and out of cells and across the barrier, which is the phospholipid bilayer. And we know from studying the fluid mosaic model of the cell membrane that there is a bilayer made up of phospholipids that have a hydrophilic head and hydrophobic tails, and they actually create a hydrophobic environment between two layers. And this hydrophobic environment here makes it very difficult for many molecules to get across. So we're going to consider how molecules do get across and whether they get across passively, that is without energy, or actively and needing energy to help them. So we'll start off by considering all the kinds of molecules that move passively. And passively means that they move with their concentration gradient. So when molecules are in high concentration on one side of the membrane, they can move without the need for any energy to an area where they are in low concentration. This is moving down their concentration gradient, and there are 3 examples here of that happening, and this is passive movement. So let's have a look at what's happening, 3 ways that this passive movement can happen. The first one that you can see here is movement without any help from any proteins. These are very small molecules that can move straight through the phospholipid bilayer without any assistance. So they have to be small and they have to be non-charged. And this is true for things like oxygen, carbon dioxide, and also for fat-soluble molecules. So things like steroid creams. They can go straight through the hydrophobic region because they are so small and they have no charge. Now, if a molecule does have a charge, or if that molecule is water-soluble, or indeed water itself, so water or ions, so charged particles, so minerals that you eat, so salts, so potassium, calcium, chloride, sodium, they need to go through a channel protein. So they're going to go down this green line here, down their concentration gradient from an area of high concentration to an area of low concentration. And their channel protein has a tube down the center that is lined with hydrophilic amino acids. So that's water-loving amino acids, which means that water and any of these ions that are surrounded by water, because they are charged, they can go through that channel without any impediment from the hydrophobic regions. There's another way that passive transport can happen, and that is through a carrier protein. And so a carrier protein still allows passive movement, but instead of the movement just being through an open hole, the carrier protein actually changes shape as the movement happens. And an example of this happening is glucose. So glucose, so I've done it in blue here, glucose, when it joins onto the protein on the outside of the membrane, that whole protein changes shape and essentially just sort of spits out the glucose on the inside. So there's never an open hole, it's a change in shape that forces the glucose through the membrane. But it's still happening passively, there's no energy involved, and it's still moving down the concentration gradient. But now we come to the last kind of transport, which is active transport. So that's what's happening here. And you can see energy is needed, and it's actually energy in the form of ATP. So this is what is created inside the mitochondria as they process the foods that we've been eating. And they release a little molecule called ATP, and ATP is actually used to change the shape of this carrier protein. So also a shape-changing protein. But this time you can see the arrow of movement is out against the concentration gradient. So we're moving molecules from where they are in low concentration, and we're moving them this time out to an area where they're in high concentration. And examples of where this happens are things like when we're trying to get ions into roots. So if the roots of a plant want to take up things from the soil, there may be very little in the soil. There may be what we might call trace elements, but they need to take them up and use them to grow in the plant, where they might be in higher concentration, but they still need them to grow. Another example might be when you're resetting your nerve cells. So nerves need the movement of ions across their membrane in order to transfer electricity and send the message. And then when you're resetting your nerves so they can send the next message, you have to move those ions against their concentration gradient. So we have active down this end and passive options without a protein, with a protein, and with a protein that changes shape. And if you need a protein, it's called facilitated diffusion, because you have to facilitate the movement. So let's have a little look at categorizing these carefully. Passive transport. Passive transport means that no energy is needed. In other words, we do not need to have any ATP involved. And the two types of transport in this category are diffusion and osmosis. They are very similar in that they involve movement down concentration gradients with no energy needed, but the definition is slightly different because the things that are moving is different. So the definition for diffusion is the movement of particles down their concentration gradient until equilibrium is achieved. And the definition for osmosis, matching in green here, is the movement of water across a selectively permeable membrane from a dilute solution to a concentrated solution. So you can see some differences in the definition here. Osmosis is exclusively applied to water movement and nothing else. We include selectively permeable membrane in the definition, whereas we don't with diffusion. Diffusion can just happen from one side of the room to another, or from one part of the blood to another, but osmosis is always about movement of water across a membrane. And this section here is about it moving down its concentration gradient, but we can't really refer to water as having a concentration. So we talk about the quality of the solution. And if it's a dilute solution, it means there's lots of water. So the water moves from where there's lots of water in a dilute solution to a concentrated solution where there isn't very much water. I'll show you what that looks like in a couple of moving images. So diffusion on the left, we're watching the orange particles mix with the black particles. There's no membranes involved, and eventually they become evenly dispersed, or they reach equilibrium, and then the whole picture resets. On the right, we have osmosis, and we have a dilute solution on this side and a concentrated solution on the right. So the concentrated solution's got lots of sugar molecules in it, and on the left it's just water. And what you'll see is down the center is a selectively permeable membrane, and the water is moving from the left to the right from a dilute solution into a stronger solution. And you actually see the levels change because there's more liquid now on the right-hand side. Our final example of transport is active transport. So we've got an image here, nice moving image of a high concentration of green particles above the membrane and a low concentration below the membrane. So this, it's just going to restart. So we've got the molecules are going through a carrier protein, ATP energy is being used, the protein changes shape to push the molecules through, and it's going against the concentration gradient. So we've got passive, active, and diffusion, osmosis, and active transport are the things you need to remember.